Hello, everyone.
Hello.
Hey,
Hey, how are you all?
good. How are you?
Still good?
Good.
We just found out like 5 minutes ago they closed schools for the rest of the year. So maybe not good.
No, not means this school year means so till the summer.
Yeah, that's a long time with my children.
True.
But I don't know what I would do if I had to be home with children right now. My dogs are driving me insane, just them. So
How many kids?
Trade your toilet paper for vodka.
I do have enough alcohol. I have plenty of toilet paper too, but please don't come rob me for that.
Oh,
It's crazy.
yeah. It's scary.
Yeah. They've never shut down the world for anything else.
Mhm.
They're really not shutting it down for this much.
Yeah, that's true. You're right. Until it's too late, probably.
Yeah.
Okay, so let's start. So today you can see my screen, right?
Yes, ma'am.
Do you
Yes.
see my PowerPoint?
Yes, ma'am.
Okay. Okay. So today we are going to start chapter 11. It talks about the microbial genetics. Basically, we are going to work on the replication of DNA, how actually DNA replicate. Then we are going to learn about protein synthesis, okay, the steps of protein synthesis. And then at the end, we will work on the mutation. That's all about this chapter. This chapter is a little long. Okay, so gene, we actually last Monday we worked on that gene. That's the segment of a DNA that actually represents a protein or code for a specific protein. So fundamental unit of heredity responsible for a specific trait, for specific protein. So sum of all types is an organism's genotype. So basically, how many genes are there together? They are make the genotype of one organism. Expression of the genotype is called phenotype. So basically, phenotype is, remember last Monday I talked about it, that phenotype is the external characters, okay? Like as we worked on that purple flower or white flower. So that flower, that external, the color, that's the genotype. But when we denote that flower color by uppercase P or lowercase p, that's the genotype or genetic combination. So this is just for the color of the flower. Okay, so we have this genotype combination for each and every character or for each and every trait. Means how tall the plant is, okay, the seed is wrinkle or round, okay. So all together, so all different characteristics, okay, so sum of all characteristics together makes the complete genotype of an organism. So basically combination of all genes together is the genotype. So expression of the genotype is the phenotype. Positively, you can understand over here, like if it is the dominant gene, then that is going to be expressed over the recessive genes. Okay, so when it's the white flower color, okay, that's the expression of the recessive gene. There is no dominant gene present, that's why that recessive genes actually has been expressed. Phenotype is determined by the specific genes within a genotype that are expressed under specific condition. Okay, all the multiple cells may have the same genotype. They may exhibit a wide range of phenotypes resulting from differences in patterns of gene expression in response to different environmental conditions. So basically, let's say the skin color. Okay, so brown versus just brown. Let's work on the brown. Okay, brown is always dominant. Now, when there is no external environmental effects, then that brown, both the genes, dominant genes will be expressed and the skin color will be brown. But the skin color actually is dependent on some types of environmental factors. Like if it is a very polluted area, okay, that pollution give you the darkness. Okay, if you stand for a long time, like if it is a really sunny area, then the skin color is going to be a little more darker. So all these things are there. For Serratia marcescens, you have seen that it makes a red colony. Okay, so if you incubate the Serratia inside the incubator, then it does not make any red color. Okay, the colony is completely white, creamish colony. But if it actually make colony below 28 degrees centigrade, like inside your bathroom shower area, then it actually make that red colony. So over here for Serratia, temperature has effects on the gene expression. Okay, so that's one factor, that temperature. Exactly same way, there are so many different factors are there that has effect on the phenotype. In that case, maybe over here in this chart, so environmental condition A and environmental condition B both has, let's say, same genotype. Okay, but because of the environmental factor over here, maybe the temperature is high in this region, then the phenotype should be completely different than the phenotype over here. So maybe this person over here will be maybe it is black color. Over here it will be a little lighter shade. So basically there are several factors out there that has effect on the genotype. Genotype is responsible for the phenotype, that's true, but some environmental condition also has effect on phenotype. So genetic information, how actually genetic information moves from DNA to our phenotype, how actually whatever we have inside our DNA, like that we actually got from the parents, okay, we inherited that information, how it is going to be expressed. The whole process is called a central dogma. So basically, whatever the information we stored or we preserve inside the DNA, our cell makes one RNA copy. Okay, it actually make a copy, RNA copy. Then that RNA from inside that genetic information, cell makes protein. So basically, the hereditary information moves from DNA to RNA to protein. Together, all these things is under central dogma. So central dogma is genetic information, movement of genetic information from DNA to RNA to protein is called central dogma. Now, when inside the nucleus, cell actually make one RNA copy from DNA, that process is called transcription. Okay, and from RNA, when cell actually makes protein, that process is called translation. So there are two different processes are there, transcription and translation. Transcription, it's like transcript, okay? Like how you did, you actually got A or you got B, so whatever you did, your score is written inside that transcript. So exactly same way, whatever the genetic information is there inside the DNA, that is being copied inside one RNA, and it happens inside the nucleus, and this process is called transcription. Now that RNA, usually cell make messenger RNA by this way. Now that RNA comes out of the nucleus and over inside the cytoplasm, actually this process takes place. So inside the cytoplasm, from RNA, cell makes the protein, and that making of protein synthesis is called translation. Okay, so clear? So together, transcription and translation is called central dogma. So our first stage is DNA replication. So the process of genetic code duplicated and passed on to each offspring. So when cell divide, so we start as a single diploid cell we called zygote, right? So what happens? So the sperm fertilizes one ova and ultimately it makes zygote. That's the first diploid cells of our body. That's the first cell. Now zygote every time multiply, and each time it multiplies and divides into two cells. Okay, so basically after cell division, so many multiple times of cell division, ultimately we grow. So because of the cell division over here, so we had just 23 pairs of chromosomes, right? Before cell divides, cell actually make its duplicate copy. So basically, before cell division, that 23 pairs of DNA, they actually make exact copy. And ultimately, there is, before cell division, there at one stage where we can find 46 chromosome in one cell. Okay, once these chromosomes are ready, then cell actually divides into two. So that's how we actually can keep the number constant, DNA number, chromosome number. Okay, and that's how we can actually transmit the hereditary information from parent to offspring. So DNA replication is very, very important to maintain the number and to maintain the hereditary information. And that DNA replication must be completed during a single generation time. So DNA replication, there are actually three different processes: conservative type of DNA replication, semi-conservative type, and dispersive type of DNA replication. In case of conservative type, so if this is the parent DNA, after DNA replication, so when DNA replicates from one DNA, we actually get two DNA, right? So in case of conservative replication, what happens? So two DNA offsprings, out of two DNA offsprings, one DNA actually is completely made up with old DNA strand. and the other DNA is completely made up with new DNA strand. So from one upspring, when we get two, one is completely old patterned, and another one, the second offspring, is completely new. Both the strands of DNA are new. That's the conservative. But in case of semi-conservative type, after DNA replication, each DNA carry one old strand and one new strand. Okay, so that's the semi-conservative type. Our DNA replication, we actually replicate by the process of semi-conservative type. Means after DNA replication, two offsprings are produced, two DNA, we get two DNA. Each DNA carry both the old strand and a new strand. In case of dispersive, it's after the replication, each DNA strand is actually the combination of both old and new strand. So it's basically the segmented, okay? And there are segment of both the old and the new version, new strands of the DNA. So Maselson and Stahl, they actually proved that our DNA replication are the semi-conservative type of DNA replication. So basically, after DNA replication, we have one old strand and one new strand. So how actually they proved that? So what they did, Maselson and Stahl, they experimented with E. coli. What they did, first they treated the E. coli with heavy nitrogen, it's 15N. Okay, so all after the first generation, all the E. coli were carrying that 15N nitrogen inside their DNA. Now, what they did, they took all the 15N E. coli and then they treat that 15N E. coli with 14N nitrogen. Now, what happened after they grow that 15N E. coli with 14N, then when they ultracentrifuge, okay, they did the centrifugation. So what happened that after treating that 15N E. coli with 14N, they actually found that there is a band at the middle between the 15 and 14N. So basically, it means like when it was heavy 15N E. coli. So after they grow the E. coli inside that heavy 15N nitrogen, all the E. coli actually was carrying that 15N DNA. Now after centrifugation, they actually got a band over here. That band, if the position of that band is here, then what they did, they took those E. coli and then they grow that E. coli with 14N. So now what they saw, like, so 14N is much lighter than the 15N, right? So there should be one 14N band and another 15N band, because 15N is heavier than 14N, so it should grow at the bottom part of the test tube and 14N should grow at the top part of the test tube. It is supposed to be like that, okay? But what happened, instead of that, they noticed that band actually got at the middle of the 14 and 15. So basically, from this experiment, they told that these band, the weight of the nitrogen or the DNA is in between 14 and 15, because this is the combination of 14N and 15N. So what happened over here, you can see, so when it was just 15N, okay, so one strand, one strand. After replication, so both were red. So all the strand were carrying 15 in. So same type of DNA was there. So we got just one strand. Now when it is 14 and 15, what happened? Let's say at the beginning it was 15. Okay, and now it is 14. So what will happen? Both the strand actually is going to get one red strand and one purple strand. So basically this is the combination of both 14 and 15. That is why the strand actually made a ring or band in between the 14 and 15. Now what they did, so that this was the first generation E. coli, they actually made second generation again with the 14N. So from this experiment, what they actually found that there were two bands. So original 14N band came out plus the hybrid band of 14 and 15. So two bands are there. So from this experiment, they actually proved that DNA replication is semi-conservative. It's not dispersive and it's not conservative. Okay, so if it is conservative, means one strand is completely, in case over here, one strand is completely new strand and the other strand is completely old strand. If this is the case, then we could have find out two strand, but we didn't see that one. So this is not the conservative type. We just had one strand, okay, in between. Now for the second generation, so from after first generation we found out that this is not the conservative, but it could be the semi-conservative or the dispersive. So in both cases the DNA is the combination of old and the new one. So when one strand is old, one's other strand is new, so they make a band intermediate between the 14 and 15. So that's why we got that purple band. So now for the second generation, what they found out after second generation, that again we saw that there are two bands. Okay, so basically that original 14 band came out because one old, one new. Okay, and one old, one new. Over here same thing happened, one old, one new. So we had this combination, right? One red, one purple. So let's say one red, and one purple. So if it breaks down into one purple, and one red, Now, so this one will go with this one. So basically two purple stand. So this, now this, so these two are together and these two are together. Okay, we have to cross with all. So purple, red. So purple and red. Now red, and so purple, purple, red, red, purple, red, purple, and we are going to get another red, red. So that's the thing happen. 2 with the combination, so 2 will make that purple, these two, and 2 will make 14. So basically that 14N and 15, 14, intermediate between 14, 15, that's the purple one, they came back for semi-conservative type, because in this case, one strand is old and one strand is new. But for the dispersive, it is the combination. In that case, we could have see just one strand, but we couldn't see that, we saw that. So from this experiment, they proved that it is the semi-conservative type of DNA replication. So DNA replication in eukaryotes. This chapter mostly talks about the bacterial DNA replication, but first we will work on the eukaryotic DNA replication. That would be very easy to understand the bacterial DNA replication. So for DNA replication, there are 3 steps. The first one is the initiation, then second is the elongation, third is the termination. So what happens during these stages? So during initiation, unwinding of the helix and the loading of the DNA polymerase enzyme complex. So DNA polymerase is the main catalytic enzyme for the DNA replication. This is the main enzyme who actually replicate the DNA. So during the initiation phase, unwinding of the helix. Remember, DNA is a double helix structure. And at the beginning, they remain highly coiled and remain associated with protein. If this is the stage, DNA cannot replicate. Okay, for that, these 2 strands has to be separated from each other. So that is why what happened, there is one enzyme, it's called helicase. That enzyme actually helped to unzip the double strand. Remember, over here inside we have nitrogenous bases, and they are actually connected, or you can say stitched together with the hydrogen bond. So helicase actually breaks down all the hydrogen bonds. So when hydrogen bonds breaks down, then that helix, so it was like this, then at certain point it gets open for the DNA replication. Okay, so that DNA helicase actually started opening the double helix from here. Now, from where that DNA helicase actually started unzipping, or you can say the point of opening of the double helix is called the origin of replication. So from where actually DNA replication starts, that point is origin of replication. And when that helicase actually unwinds the DNA double helix, it looks like a bubble-like structure. So this bubble-like structure is called replication bubble. Okay, and in each replication bubble, that Y-shaped area, there are 2 Y-shaped area over there and over here. These 2 area is called replication fork. So in one replication bubble, there are actually 2 replication fork. So once that these two strands are separated from each other, then polymerase enzyme can start replicating the DNA. So next stage is the elongation. So sequential addition of deoxyribonucleotides to a growing DNA chain. So now what happened? That DNA polymerase, it actually start reading the DNA polymerase. This enzyme actually has a one excellent character. It can read the DNA nucleotide sequence over here. It can read DNA nucleotide sequence, and at the same time, it can add new nucleotides. So it actually read and start adding the nucleotides, and that's how it actually makes a copy or new DNA strand by copying the other one template strand. Okay, so these two strands now are called template strand. Means the new DNA strand will be complementary of these two template strand. So that's the elongation part. Termination, when DNA is completely copied actually. So these are the three steps have occurs during the DNA replication. So this is the big picture over here. Records the actions of 30 different types of enzyme. First, separate the strands. Each strand act as a template. So this is the double helix structure. Helicase enzyme unzip the double helix. Okay, it separates both the strand, and each strand is called template strand. Template strand means these two strands are going to be copied. Okay, that's why they are the template strand. DNA replication actually happens from 5' to 3' direction. So this is the direction of DNA replication. Very, very important. One old strand pairs with a new one and produce two new DNA molecules. So over here you can see in this picture, so what is happening? So helicase enzyme first unzip. Once it unzip, then it creates one replication bubble. Okay. For eukaryotic chromosome, our chromosome is really long. So inside our chromosome, we can have multiple replication bubble. Okay, so it looks like this for eukaryotic replication DNA. Now the DNA polymerase, it's DNA polymerase 3, that's the main enzyme that catalyzes the reaction. This enzyme now start reading the template strand. So over here, you can see there is one polymerase enzyme for each strand. Okay, and every time DNA polymerase can make new strand from 5' to 3' direction. So what happens? Look, if this is the DNA strand, let's say this strand goes from 5' to 3' over here, then this strand should go from 5' to 3'. Last week we worked on that these two strands actually runs in opposite direction, and that is why they are the antiparallel. Do you remember that?
Yes.
Okay. Yes. So that is why, so if one strand is 5' to 3', then other strand must be 3' to 5', right? And what happens? So if this is the template, and it has all the nitrogenous bases. Let's say it's adenine, thymine, adenine. So that's how all the bases goes over here. Now that polymerase enzyme over here, it actually start reading the nitrogenous bases, and it picked up that nucleotide. So all the DNA nucleotide like adenine, guanine, cytosine, and thymine, all the nucleotides already are there inside the cell. Okay, but before DNA replication, cell actually make ready all these different type of nucleotides. But somebody has to read the direction, the sequence of the nucleotide, okay? And it has to bring all those, or you can say it has to arrange all those nucleotides, okay, along the strand. So that's what the DNA polymerase does. So it actually read. So over here, let's say we have A. So complementary, it should be T. So DNA polymerase actually pick up thymine, then adenine, then thymine. Let's say over here cytosine, then it will pick up guanine. If it is guanine, then it will pick up cytosine. So that's what DNA polymerase enzyme. So this is DNA polymerase enzyme. So DNA polymerase picked up all the DNA nucleotides and it arranged over here. So that's how it actually make a complementary copy. Now, so all these are loose, right? Then we have a DNA ligase enzyme. It's called ligase. That enzyme actually stitched all these nucleotides together, and that's how we actually get a complementary DNA strand. So that's the whole scenario, big picture of the DNA replication.
Miss,
Yeah.
Miss Nandi, it is like if one of the sides was the DNA and the other one was the RNA.
Yes, same thing, but RNA we have just one copy, one strand, DNA strand. Yeah, but the same procedure.
Okay. Okay.
That's what you asked, right?
Yeah, I was just wondering if the one that is the copy is the RNA. So yeah.
Oh, no.
No?
This is not the RNA. This is not the RNA. We are talking about DNA replication. So from one DNA, we are making two DNA.
Okay.
Remember before cell division, I told that over here, I told that before cell division, so we have one cell, right? And we have 23 chromosome. When cell is going to divide and it will make two cells, how are we going to divide the DNA? So before cell division, that DNA, the 23 pairs of DNA, they actually replicate.
Okay.
Okay, they make their exact copy, and that's how that from one cell we can make two cells.
All right.
Okay, so that's the DNA replication. So making copies of DNA. Okay, and here we are actually working on how DNA replicates or how DNA make copies.
So before the replication, we had 46, right? Before replication, right?
Immediately before cell division, at certain point, we have 46 chromatids. We don't have 46 chromosome. We don't say chromosome. So basically what happens, so let me go back over here. Okay, so over here, we had, let's say, this was the chromosome, right? Okay, now, so two strand, right? So each strand make their own copy. Okay, these are the template strand, and so they make their own copy. So basically from one DNA we get two DNA. So at the beginning we had one or two strands. Now we have 1, 2, 3, 4 strands. all 4 strands, they remain attached at the middle part. Okay, this part is called centromere. So basically, 2 pairs of DNA remains together, attached at the centromere area. When they are attached, actually we have 2 DNA, but as they are attached, we don't call them DNA that time. Okay, so but they remains like that way. So if you count 1 and 2, yes, we do have 46. Okay.
Okay.
Okay. Okay, so big scenario, what happens? At the beginning, the strands was highly coiled and attached. So with the help of helicase enzyme, these 2 strands get separated from each other and they make a bubble-like structure, and this is called the replication bubble. Okay. Now, the point from where actually DNA strands get separated from each other, or you can say the point from where DNA replication start, that is called ORIC, or it is called origin of replication. So from origin of replication, helicase enzyme starts working, starts separating the DNA double strands, okay, and it makes a replication bubble. In each bubble, there are 2 Y-shaped area. Each area is called replication fork. So in one replication bubble, we have 2 replication fork. Now, when it's ready, then DNA polymerase enzyme, polymerase 3 enzyme, it start reading the DNA sequence and picked up the DNA nucleotide and arrange along the DNA strands, the line. So, and ultimately at the end, DNA ligase, it stitched all the DNA nucleotides together, and one complementary DNA strand is going to produced. Clear?
So where does the DNA polymerase will go and get the reading for the nucleotide for it to attach?
Oh, it's inside the cell. So the whole thing, look, that chromosome. So these are the chromosome, right? They are inside the nucleus, right? So this is our nucleus. So we have that all those DNA nucleotides over here, that adenine, guanine, cytosine, thymine, all these nucleotides remain over here. They are actually predisposed. Before cell division, cell actually make all these ready.
So the DNA polymerase will not only read the nucleotide, but also make copy of them. That's what you mean?
No. It does not make copy. Already there. It's already there. These adenine, guanine, cytosine, all these are here before DNA replication.
Okay, they are inside the DNA or in the cytoplasm? I mean, they are inside the nucleus or in the cytoplasm? That's my question.
Nucleus, yeah, they are here. Like before cell division, what happens? The cell actually increases in size. Why? Because it actually store all these things. It actually make ready that adenine, guanine, cytosine, thymine, all different enzymes, that DNA polymerase, DNA helicase, all these enzymes remains ready inside the nucleus before the DNA replication starts.
Okay.
It is already there. But somebody has to read the direction, right? That thing is done by the DNA polymerase. So DNA polymerase read the direction, and accordingly it pick up that DNA nucleotides. So basically, as I told, over here, let's say at the first one, it is adenine. So on the complementary strand, it must be thymine, right? So thymine nucleotide has to be there. So who will do that job? Who will bring that thymine over here? So that is done by DNA polymerase. DNA polymerase read that, okay, so over here at the first position, it has adenine. So that means the complementary strand must have thymine. So it pick up thymine from the nucleus and it placed it over here. Then the next is thymine. So it has to be adenine. So DNA polymerase pick up that adenine and it places the adenine over here. And that's how it actually make the complementary strand, DNA strand. And then all these nucleotides are stitched together by enzyme ligase.
So the nucleotides are just free floating and the
Exactly.
okay.
They are just free floating and they are already there inside the nucleus.
Okay. Thank you.
Okay.
All right, thank you.
Okay. Okay, so now over here, the whole scenario. Now we will go little deeper. Okay. So what happened? At the beginning, the DNA was highly coiled, right? First, somebody has to make it straight. There are gyrase enzyme, or it are also called topoisomerase enzyme. This enzyme actually uncoiled the DNA and make it almost straight. It's not straight, but it's like spring-like structure. Still, it's highly coiled, but it's almost straight. So that part is done by the gyrase enzyme. That's our first enzyme. Now, second enzyme is called single-stranded protein. That is another enzyme. What it does, it actually put a load over here at the end of the spring. So if you add a weight at the end of the spring, then the spring becomes uncoiled and it's almost becomes straight, right? So that thing is done by single-stranded protein. So now the DNA strand is completely almost straight, but still they are together. That part is done by the helicase enzyme. So helicase is our next enzyme. Helicase unwind, it actually breaks down the hydrogen bond. Okay, and that's how it actually separate. So basically it was like this and some area this. So it separate the little portion, not the whole strand, not the entire length is going to be separated, but just a little portion is going to open at a time. Now DNA polymerase enzyme, okay, comes into the scenario. So our fifth enzyme is DNA polymerase. What it does, so it actually read the DNA sequence and it pick up the DNA nucleotides, adenine, cytosine, guanine, thymine, okay, and then it actually arrange all these nucleotides along the length of the template strand. So that's the function of DNA polymerase. Now, DNA polymerase only can add up nucleotides at 3' end. 3' end, there has to be one hydroxyl. So basically DNA polymerase cannot add up all this nucleotide from the scratch. It only can add up if there is any short segment of either DNA or RNA is there with 3' hydroxyl end. Then only DNA polymerase can add up all these things. Are you clear at this part?
Hello, I have a
I'm
question.
not really clear on that part.
Are you clear up to 5?
I was, which one is the fourth? I don't see any name under the
Fourth I'm coming later.
Oh, okay.
Okay, but till up to this, did you get it?
What is the name of the last one? It says DNA / what else? What's the rest on the bottom of it?
DNA or RNA?
DNA, RNA, and the 3, what is the 3, what?
3' hydroxyl group.
Okay.
So DNA polymerase, it actually read the DNA sequence, it pick up the DNA nucleotides, and it actually arranged serially. Okay, but it only can start doing that if there is already a small piece of DNA or RNA is there. Then only it can do all these things. It cannot start making DNA strand from scratch if there is no DNA or RNA portion. So that problem is actually going to solve by RNA polymerase enzyme. What it does, unlike DNA polymerase, it does not need any small DNA or RNA segment. It can actually make a very small segment of RNA. If a small segment of RNA is there, then DNA polymerase can start adding up all the nucleotide. Okay?
So in other words, the RNA polymerase, that was the one that carries the message for the DNA polymerase to pick up. That's what you mean?
No. So basically what happens, look, so 2 strands were there, right? 1 strand, and this is 2 strands. But these 2 strands were highly coiled. So these are the enzymes, they separate the strands from each other. They try to make both the strands straight, okay? And then they separate both the strands. So now at this moment, these strands are separated from each other, and these are the template strands. Okay, so are you clear up to this?
Yes.
Okay.
Okay.
Now you understand that over here, all these strands has that nucleotide sequence, right? That are nitrogenous base.
Yes.
So to make complementary strand, what we have to do, if this is one DNA strand, okay? So it has adenine, then thymine, cytosine, guanine, and adenine. If I ask you, and this is 5' to 3', if I ask you to make one complementary strand, so what would be the sequence?
So the DNA polymerase will read the adenine and
No, I'm just talking just in general. So over here it should be thymine, adenine,
G, D,
C, right? And this should be 3' and this should be 5', right? Okay, so to make complementary strand, what you did, you actually first read it, right? So we had A, you read that. And then you told that, okay, so if it is complementary, then we must have T, thymine over here.
Yes.
Correct?
Yes.
Same thing is done by the DNA polymerase. So DNA polymerase first read this strand. Okay, so adenine is here. So we must have thymine here. So it actually pick up the thymine for this area. Then it reads the next one. Okay, so thymine, that means it actually picks adenine. And that's how it make one complementary strand. Are you clear now?
Yes.
Okay, now to do that, so basically what it is doing, it is reading and at the same time it is actually picking up. So all the adenine, thymine, guanine, so let's say 100 guanines are here, 200 thymines are here. So it is just picking up one thymine and adding over here. To start this job, for DNA polymerase, it cannot just start if there is no DNA or RNA segment already there. So if there is a small piece of DNA or RNA is there, then only DNA polymerase can start this job. Okay, so that small piece of DNA or RNA we need to start the DNA replication. So that problem is going to be resolved by the RNA polymerase enzyme. What it does, before even DNA polymerase start working, it actually make a small RNA piece. In one small piece, there should be 10 to 20 nucleotides. Very small RNA, it actually makes and it places it over here. And now this small RNA is called RNA primer. So basically to start DNA replication, DNA polymerase need a small RNA primer, means a small piece of RNA it needs.
Is that the RNA polymerase?
No, that is done by RNA polymerase.
Okay.
Okay, are you clear?
So the RNA primer is made up by the RNA polymerase. Okay.
Yeah. So if RNA polymerase is here, so this is one DNA, let's say adenine, cytosine, guanine, thymine, adenine. This is the sequence, okay? Now, if RNA polymerase is here, that's a small piece of RNA, and that is made up by RNA polymerase enzyme. If this is called primer, RNA primer. When the primer is here, then only DNA polymerase can add up all these nucleotide. Okay, so that first nucleotides remain attached with the RNA primer. Are you clear now?
Yes.
Okay, so now that is why DNA polymerase actually can make DNA new strand that goes from 5 prime to 3 prime. I hope you remember that nucleotide structure. One nucleotide goes like this. Do you remember? Nitrogenous bases are over here. Okay, this is number 1, number 2, number 3 carbon is here. Over here we have one OH group. This is number 4, and 5 carbon remains over here, and here we have phosphate. Okay, now for the second nucleotide, so this is OH over here, nitrogenous base is here, this is the 4th carbon, and phosphate is there at 5 carbon. So when they make that phosphodiester bond, so how actually that nucleotides are attached with each other. So what happened? That 3-hydroxyl group remains attached with the 5-phosphate. So this is the 3' group, remains attached with the 5' group, and they make that phosphodiester bond. And that's how two nucleotides remains attached with each other. So basically what happens for DNA polymerase, it must have a free OH group. So if there is one free OH group, then only DNA polymerase pick up the nucleotide and it actually add that 5-phosphate with that 3-OH group. So that 3-OH group, for the 3-OH group, RNA polymerase make a small piece of RNA that has at the end, it has that OH. So basically it starts from 5' and this is the 3'. So this is the 5' group and end, and this is the 3'. So every time DNA polymerase can make new strand that goes from 5' to 3'. So every time DNA synthesis goes from 5' to 3' direction. Are you clear?
Yes.
All of you?
Yes, ma'am.
Yes, ma'am.
Yes.
Okay, so now just try to consider these two strands, okay? So this is one strand. You see this strand is going from 5' to 3'. So this is the 3' end and this is the 5' end for this strand. So complementary strand. So this strand, the lower strand goes from 3' to 5'. Are you clear?
Mhm.
Okay.
Yes.
Okay, and this is
Yes.
the origin of replication. Okay, so opening up, you can see that this part is coiled. So opening up of the coiled section actually started from here. So it started open from this part. Okay, this is the origin of replication. So for this strand, so let's say this part was closed, okay? Only this part was open at the beginning because it opens very slowly. So it goes, you know, slowly this chain is going to open. So this part was completely closed. So let's say that only from here to here was open. So what happens? For this strand, okay, it is somebody's microphone here. So Esmeralda, could you please turn off?
I'm sorry. Yeah, I'm sorry.
So from this, for this strand it's 3' and 5'. So its complementary strand should be 5' to 3'. And every time DNA polymerase only can make DNA strand from 5 to 3. So for this strand, DNA polymerase will start copying DNA nucleotides here and it will start making the new DNA strand and it will go towards the replication fork. So in this case, the DNA replication is continuous. It will start from this point, okay, from the beginning, and it will slowly, it will go towards the fork. Now this portion will unzip and that strand will continue. Okay, so slowly it will open and the process will go on ultimate and all the way to the end. So for this one, there is no problem. So DNA replication goes continuously, and that is why this strand is called leading strand. Okay. Now imagine this part. For here, this is 5' and this goes towards the 3. So its complementary part is 3 to 5, but DNA replication only occurs from 5' to 3'.
Yeah.
Right? Opposite direction it cannot go. So in this strand, what happened? DNA replication actually it's not continuous. So what happened? So the DNA polymerase actually start from 5' end and it goes this way. It makes a small segment this way. Then it jump over here, and from 5 prime to 3 prime, make a small segment, and again it jumps over here, and from 5 prime to 3 prime, make a very small segment. So basically, on the other strand that goes from the 5 to 3, 3 to 5, for there, DNA replication is not continuous. In this case, DNA polymers make very small segments, and each segment is called Okazaki fragment, okay, and in opposite direction. So all the small fragment ultimately then is going to stitched together by DNA ligase. And in this case, DNA strand or DNA replication is not continuous. So that is why this strand is called lagging strand. So DNA replication on both the template, on both strand, goes in opposite direction. One strand DNA replication is continuous. It goes from 5 prime to 3 prime direction, and it goes towards the replication fork. And that strand is called leading strand. On the other hand, on the other side, DNA replication is discontinuous. It makes a very small segment at a time, and all those segments later become stitched by the enzyme DNA ligase. That strand is called lagging strand, and in this case, DNA replication goes away from the replication fork. So this side, replication was going towards the replication fork. This is the replication fork. This side, replication was going away from the replication fork. And this is called lagging strand. This is called leading strand. Over here, all these small segments are called Okazaki segments or Okazaki fragments. It's actually from the scientist, husband and wife, scientist Okazaki, they together make. So to show the respect, all these segments are called Okazaki segment. So every time DNA replication goes from 5 prime to 3 prime. Over here, yes, it's a long strand, but it's still from 5 to 3. Over here, yes, it's still from 5 prime to 3 prime direction. Are you clear?
Yes.
Yes.
Did you all get it?
Yes, ma'am.
Yeah.
Yes, ma'am.
It's little complicated, but once you get it, then it will stay forever. Okay, so I already talked about everything in this slide. Okay, for bacteria, same thing happened. For bacteria, it's very quick actually, DNA replication. You can see that for E. coli has 4.6 million base pairs. So they have bacteria, they just have single circular double-stranded DNA. Okay. In one chromosome, they have 4.6 million base pairs. But to replicate, so it just take approximately 42 minutes. So quick. Okay, so for us, for our, that for eukaryotic chromosome, it's just straight, right? We have linear chromosome. So that's how it looks like this. But for bacteria, they have a circular chromosome, okay? So what happens, so this is the origin of replication, or it's called ODIC. So from here, this is the point from where both the strands actually started separating. In this case, the DNA replication is bidirectional. So one replication goes this side and another replication goes this side, and for both the strands. Okay, and ultimately once these two directions meet, and that point is called tar, where they end up. Okay, when they meet at tar point, both the direction, and then they actually stop the DNA replication. So it starts from the ODIC, origin of replication, and it ends up at tar. So you can see over here, this is the big picture. So what happened, let me move it here. Okay, so in this picture, you can see the last one over here. So this is the origin of replication, okay? They opened up over here. And that point over here, from here, it was 5 prime. And if I say that this is the end, then this is the 3 prime. Okay. Now for this side, this is the 3 prime, and this is the 5 prime. So you can understand, like, if this is the point, if this was the 5 prime, then positively this should be the 3 prime end, right? So when this side, so basically 2 DNA replication will go on, one this side and another this side. Okay, so that is why it's bidirectional. Now just consider this side. This is 5 prime end, okay, and this is the opening. So it should start from here. So 5 prime end means complementary strand should be 3 prime. But for DNA replication, every time it needs to start from 5 prime. So in this case, it will be lagging strand. Do you see? This is the Okazaki segment. So if this is that, then it's opposite. Over here, it's the leading strand, continuous. Okay? But in this side, so it was 5 prime, means this is the 3 prime, this is 5 prime. So in this case, complementary strand from 5 prime. So this portion should be leading strand, and the lower portion should be the lagging strand. So direction is this way, but for this one, direction is that way. Completely opposite direction this way. And for this one, direction that way. Are you clear? But ultimately, if you consider the whole scenario, this is the whole direction, but this is the direction. Are you clear?
Yes.
Yes.
It's little complicated.
Yes, ma'am.
Okay, so do you have any question? So this is the whole scenario. Okay, we are going over it again. So helicase enzyme, what it does, it unzip the DNA helix. Means it actually breaks down the hydrogen bond. In between the nucleotides. This hydrogen bond is going to break down by helicase enzyme. Primase, primer. So this is actually RNA polymerase enzyme. This RNA polymerase, what it does, it makes a small RNA segment, which is called RNA primer. Small means very small. It's actually 10 to 20 nucleotide sequence. DNA polymerase 3, it actually read and add all the bases. At the same time, it also proofread. Okay, if there is any mistakes, it actually stop working. Now, DNA polymerase actually remove the primer. So once that DNA replication is done, who is going to pick up that RNA primer? It will be there, right? So new strand over here, it's complete, but at the beginning, there is an extra RNA segment. Okay, who is going to pick it up? That DNA polymerase 1, that's the enzyme, it actually removed the primer. Ligase enzyme, it actually stitched all the DNA nucleotides together. Topoisomerase 1 and 2, they actually control the supercoiling and untangling. Clear? Make sure you read. You have to memorize which enzyme works what. Okay, so for eukaryote, you can see we have a linear chromosome. So for us, multiple replication bubbles are formed. But for bacteria, you have seen just one replication bubble is there. Okay, because they have a circular. Okay, so the telomerase, okay. So
It's time.
it's time. Oh, okay. Okay, so let's keep it up to this. I will start from here then. Next Monday. Okay.
Thank you.
Thank you.
Okay, make sure to read the text.
Thank you, Dr. Mandy.
You're welcome.
Thank
Have a good night.
Okay, have good weekend.
Bye, everybody. Stay safe.
And be safe.
Bye. Bye.
Bye.
Yes, it is. That's my Monday, Wednesday.